Hello, another video for you, and in this video we are looking how genetic engineering is done. Okay, so we will take one example of how it's done. For example, we are going to modify bacteria to produce human insulin. Okay, so in the previous video I talked about how genetic engineering involves transferring a gene from one organism to another. So let's see how it's done. So number one, we have to isolate, in this case, we isolate the human insulin gene because we want this gene and this gene makes insulin. So how it's done? Normally, we will take cells from the pancreas. We have to remind ourselves that the organ pancreas, exactly the islets of Langerhans, that's where you have those special cells that secretes the protein insulin. Okay, insulin is a hormone, but let's not forget it's also, okay, a protein. Insulin is a hormone, okay, but it's basically a protein. And that protein is coded by a gene. So what we do, we take the cell, we break it down. Okay, you don't need to know the detail of the process. We break down these cells, we dissolve all the organelles, and we ultimately want to precipitate the DNA. So we add some chemicals, and ultimately we get the DNA. All right, so we're basically extracting DNA. So once we get the DNA, this is where we use an interesting enzyme called restriction enzymes. Okay, restriction enzymes. Now, what these enzymes they can do, think about restriction enzymes as some kind of molecular scissors. So what they can do is they will cut the DNA. You have to remember, DNA is a double-strand molecule. So they cut the DNA at specific sites. So restriction enzymes are specific. So scientists, we know the sequence of the gene for insulin. So that would be, let's say, the insulin gene. So we use restriction enzymes, and these restriction enzymes, they cut the particular piece of DNA that we're interested in, basically the gene for insulin. Now this gives us something like this. You get a piece of DNA, okay, with a slight, like with a few bases that extends from it. This creates what is known as the sticky ends. Okay, so when we're cutting that piece, the gene from the DNA, the restriction enzyme cuts it in such a way that you get sticky ends. So this is basically how we have isolated the human insulin gene. Okay, so we use restriction enzymes. The next step is we have to have a look at the bacteria, because the aim is to modify a bacteria. So you basically want to insert this particular gene in this bacteria. That's where you need to know something about plasmids. Now, interestingly, bacteria, they have plasmids. Now, plasmids are basically, they have their own DNA. You can see it here. But they also have rings of DNA that's called plasmids. And interestingly, plasmids can be modified to include genes from other organisms to form recombinant plasmid. That is, once you've added another gene to this, you get something called a recombinant plasmid. Okay, so in step 2, we basically use the same restriction enzyme to cut the plasmid DNA. Okay, above here, we've used restriction enzyme. We know they're super specific. They cut only at specific, points on the DNA, at specific sequences on the DNA. We use these same enzyme, and what we do, we extract the plasmid from the bacteria. All right, we use the same restriction enzyme. Okay, chop, chop, we cut it here. And what you see, once we remove that piece from here, okay, you also have sticky ends on the plasmids, right? Now, guess what? Just like a puzzle, what we're going to do, we've managed to cut the plasmid and we have sticky ends with the same restriction enzyme. We already have a piece of the human gene, insulin gene. What we do? Third step, the pieces of human DNA. Okay, of course, we do this on many plasmids and we make sure we get a lot of human genes. The pieces of human DNA, basically the insulin gene containing the insulin gene, are joined to the cut plasmid using ligase enzyme. Now, so far you know restriction enzyme, they're like the scissors, they cut the DNA. What basically we do, we take the insulin gene, okay, and we mix it. Okay, all of this, boys and girls, happening in a test tube. So we mix it, and basically what's going to happen, the sticky end of the insulin gene, okay, the sticky end here, will bind complementarily to the sticky end of the plasmid. So ultimately it fits like this here, and we need some kind of between quotes, glue. So something needs to stick them together, and that's where ligase enzyme comes into the picture. So we, to that same test tube, okay, we had the plasmids, right? We had the insulin gene. We also add the ligase enzyme, okay? And it's all happening in the test tube. We wait for it, and these ligase will seal, okay, these sticky ends together. There's a word for this. They say anneal. They anneal the sticky ends together. And boys and girls, once this happens, you get something called a recombinant plasmid. All right. So this plasmid here has been engineered. You have the human insulin gene in it as well, as you can see just here. Okay. Now the next step is basically the plasmid. It acts as a vector, putting the human insulin gene into the bacterium. So we're gonna take that recombinant plasmid. Okay, there's a new word for you. We describe this as a vector because it's kind of carrying now the human insulin gene into the bacterium. So we basically do this. Obviously, there are a lot of steps in doing that, but like just to make it simple, we take the recombinant plasmid and we insert it into the bacteria. Just like this. It's not magic. It's science. Once the bacteria gets that recombinant plasmid, is going to use its protein producing components like organelles like ribosomes and stuff, and ultimately once the recombinant DNA is there, plasmid is there, human insulin will be secreted. Remember, okay, this whole thing, DNA gives mRNA gives protein. So if you have the gene for insulin, the bacteria is going to transcribe and translate the insulin gene, and you get human insulin is made. Fantastic. So this is basically a genetically modified bacteria. All right. Now, once we get this, we have one bacteria. What we can do, last step, the genetically engineered bacteria are grown in fermenters. Remember the fermenters? We put them, these bacteria in there, those GM bacteria, and we let them multiply. We make sure the conditions are perfect, and they're going to make insulin coded by the human DNA. So those bacteria will reproduce asexually to form large population, and in these fermenters, after some time, there will be a lot of insulin, and then we can separate and filter out the bacteria and get the human insulin, and ultimately we can put them in vials and distribute commercially and save lives. That's it. That's one amazing example of how genetic engineering is done. So I suggest you go through the steps again. Okay, you also check your notes. It's all gonna make sense. All right. If you have any question, please send them to me. Take care. Bye.